and the laissez pointer is the red one.
Okay. Thank you very much, everybody, for attending this year's symposium. I'm going to present the work I do here at the College of Marine Science. So my lab is mostly involved in fish studies, and we have a fish egg monitoring that is coming up. It's going to be long-term on the West Florida shelf, and it will start this spring. We will have to interpret some long-term trends, and for that, we need to understand and have knowledge of whether fish species will retain or export their eggs off the continental shelf. More specifically, if, for example, we have a decrease in the temporal trend, then it can have different types of implications. It has more important implications if, for example, the species retains its eggs on the shelf. If the eggs are exported on the shelf, then the implication is not as serious. So our approach is to first locate the actual spawning sites, and I will elaborate on that later. And once we have the spawning sites, we can numerically model all the trajectories of the eggs from those known spawning sites. We will do some forecasting and high casting. So to locate those spawning sites, there are three approaches. The first one is to find the adult females that are about to spawn. The second one is to find the females that have spawned within the past 24 hours because the eggs didn't have time to move too much in the water yet. And the last one is a combination of DNA barcoding and some hindcasting, but I will not discuss this in this presentation. And you probably learned a lot about DNA barcoding with Jeremy's talk this morning. So on July 11th of 2017, we had a great sampling effort. We had 6 commercial boats and 2 FIO research vessels leave from Panama City and from St. Petersburg to go sample on the West Florida shelf. We collected 520 adult fish during the day, and we collected drifting eggs the following morning in those hexagons on the map. So we were able to catch fish in those specific locations, and that's from where we will simulate the trajectories of fish eggs. Of our 520 fish, the most commonly encountered species were red snapper, sand perch, and vermilion snapper. And those fish are reef-associated fish. So let's talk about the first approach, and that consists in finding the females that are about to spawn. To do that, we look at the fecundity, and we have to define a few words here. Fecundity is the number of eggs that are ripening between the current and the next spawning season. And what we're interested in is the batch fecundity. That's the number of eggs a female can produce in one spawning event. And this definition is valid for batch spawners. Those are fish that spawn several times in one spawning season. And to look at fecundity, we look at ovaries that you can see on this image. Speaking of ovaries, those can be macroscopically staged from immature to spawning imminent. And as you may have guessed, we are interested in the latter. And that's because they present hydrated eggs. Once we identify the stage, especially the spawning imminent that look like this, we can wash out all the eggs from the ovary with hydraulic pressure and preserve them in a little container. When we're back in the lab, under a sterile microscope, we can distinguish between hydrated and non-hydrated oocytes. So to give you a little bit of explanations, oocytes in the ovary will start hydrating. It's a process that occurs within a few hours before the spawning event. And that's how they look. Non-hydrated are small and opaque, and hydrated are large and clear. So once you see hydrated oocytes, you know that the female is about to spawn very soon. We went a little further in this fecundity work, and we estimated the batch fecundity. But a batch can range from just a few eggs to hundreds of thousands of eggs, and it's not really realistic to count all of them. So what we did is a volumetric estimate. It consists in suspending eggs in a known volume of fluid. Then we homogenize the egg distribution, and instead of counting all the eggs in the whole samples, we only count hydrated eggs in subsamples, in several subsamples. Once we get that, we can extrapolate the numbers to the entire sample. So that concludes our first approach. For the second approach, that consists in finding the females that have spawned within the last 24 hours, we also use ovaries because they come in pairs and we preserve the second lobe. We did very thin sections that we mounted, ovaries of course, and we mounted them on slides. That's how they look after. And under a microscope, you can determine the presence of post-ovulatory follicles, also known as POFs. You can also age those POFs. Here's a photomicrograph of oocyte within an ovary. You can see different oocyte stages. This is a hydrated egg, and this is the POFs that we're looking at. The age is very important because the recent POFs indicate recent spawning, and they also indicate the spawning sites. So if we look closer, we can distinguish the age. For the recent POFs, they look like this. They are round shape. They have a cavity in the middle. That's because they have released the eggs that has ruptured the follicle and released the eggs. Those POFs are under 24 hours old, and they indicate that the female has spawned recently. Older POFs look like this. They are shrunken. They don't have a middle cavity, and that's because they have started being reabsorbed by the ovarian tissue. And so to be able to find the spawning sites, we don't use the older POFs. We use the recent ones. So what did we find? This is the map that I showed you earlier with the adult collection sites. And after our fecundity work, we determined the actual spawning sites. And from those, we will simulate the trajectories of the eggs. We went a little further in this fecundity work and were able to get some reproductive parameters. As a reminder, we went fishing between 11 a.m. and 8 p.m. And whenever we plot the collection times, we were able to define some spawning time windows. For red snapper, it was pretty well defined. You can see a start and end time. And for vermilion snapper, we have a start time, but we stopped fishing at 8 p.m., so we're not sure if they continued spawning after this time. For sand perch, unfortunately, we were not able to find a window. That's because all the females had recent POFs. And so they have probably started spawning before 11 and stopped spawning after 8, but we don't know for sure. A few more data on the spawning females. Because I did the estimate of batch fecundity, we found that there were 100,000 eggs per batch for vermilion and 80,000 eggs per batch for red snapper. I want to bring your attention to the size of the fish. The vermilion was smaller, but had a larger batch, and the snappers was larger, but a smaller batch, and that's not uncommon for fish. The batch fecundity was not estimated for sand perch because it would have been biased. When we did the POF study, we found that some eggs have already been ovulated. Now that we have all the locations and some extra parameters on the reproduction, we can investigate the retention versus export of those eggs. What we're going to do is use the West Florida coastal ocean model in collaboration with the Weisberg lab. And those models allow us to track where the water came from and where it went to. For our specific questions, we're going to forecast the egg and larva movement after the spawning. And we will hindcast the location of spawning sites from the eggs that were collected via plankton net. So this research has already been done in the past. The approach has been applied, and that was done on larvae and juvenile gag transport by Weisberg and others. Here you have two series of images. On the left are surface particle trajectories, and on the right are near bottom particle trajectories. So the models allow us to simulate the trajectories of eggs at different depths. And the idea here was to determine if the bottom waters or the surface waters were more influencing the transport of the larvae and juvenile from the spawning sites to the known juvenile settling sites. So what we're hoping to do is to apply this approach during our shelf monitoring project and apply that to other species. So in conclusion, we were able to locate definite spawning sites, and we can now input them into a numerical model to determine if there was retention or exportation of the eggs. And we also were able to obtain other reproductive parameters for red snapper and vermeil snapper. And especially for red snapper, that is a commercially important fish. I would like to acknowledge all my funding sources and thank all the people who are supporting me in this journey. I'd like to thank you all very much for being here, and I will take questions. Thank you.